A very good evening to everyone. Today, we from Group 5 would like to present on the histopathology workflow of a specimen. The specimen that we have chosen for this workflow is the liver tissue sample from human. But first, allow me to introduce myself and my teammates. I'm Thashwina and we have Kaveeyarasi and Thivayta Shanmugam here. Histology is the study of the anatomy, structure, and function of tissues. After tissues are removed from the body, such as a suspicious mole, a burst appendix, or even cancer, they must be prepared and processed so that they may be examined under a light microscope on a glass slide. The first step of the histopathology workflow of liver sample is the specimen logging. The specimen is documented by assigning it a lab number that is unique to the particular patient and will identify the specimen for the patient in the histology laboratory once the patient's details have been recognized with their name, date of birth, and identification number. After logging the sample, we must perform a physical examination to characterize and measure the liver tissue. A piece or a segment of liver tissue was dissected with a scalpel or blade to acquire a sample of liver tissue. Some of the liver tissue is taken out and put in a cassette where it is fixed with a 10% formalin aldehyde.
Okay, my name is Kaveeyarasi and I'm going to explain about the tissue processing, embedding and sectioning. So the next process was tissue processing, which describes the steps required to take tissue sample from fixation to state where it's completely in filter with the suitable histological wax and can be embedded ready for section cutting on microtome. So the instrument that we use for this process is a automatic tissue processor. So basically, tissue processing contains the four basic steps, which are dehydration, clearing, infiltration, and impregnation. So at first, the sample is dehydrated using an alcohol solution for an hour to remove the water. And for the clearing part, the tissue is soaked in the clearing agent. The basic agent we use is xylene, is to remove the alcohol for an hour. And after that, the tissue will be immersed in the liquid paraffin wax solution to remove the clearing agent and allow paraffin to diffuse into the tissue for an hour and let the paraffin to saturate the tissue for overnight before the tissue undergoes the embedding process. So the next process will be embedding. So the instrument we use for this process is a paraffin dispenser unit. So the process is basically start with tissue sample is removed from the cassette and placed into the mold. The mold is filled with the liquid paraffin wax using the paraffin dispenser unit. And after that, the higher temperature keeps the paraffin wax as a liquid, so it is easier to manipulate and the tissue sample from cassette to the mold. And after that, the tissue block gives the tissue a support and shape for the sectioning. And the next part will be the sectioning. And the instrument that we use for this part is a rotary microtome. So basically, this procedure got three basic steps, which are trimming, sectioning, and fishing. So trimming is basically cut the unnecessary parts of block by shape of the edges. And after that, the sectioning, like cut the very thin section that are cut off from the ribbon from the 5 to 4 section of the tissue. And after it the being sliced, the paraffin ribbon is floated on the water bath about 60 temperature to prevent and wrinkles. And after that, the fish moves to the fishing process, which is the section can be then picked up from the water bath and placed on the glass slides.
Hi, my name is Thivayta and I'm going to discuss on the following histopathology workflow, which is HNA staining. A common laboratory method that uses two dyes called hematoxylin and eosine that makes it easier to see different parts of the cell under a microscope. These stains are frequently used in laboratory to highlight the structures in biological tissue for viewing with the aid of different microscopes. It is also used to define and examine bulk tissues by highlighting the muscle fibers or connective tissues, and also cell populations by classifying different blood cells or organelles within the individual cells. Once the section is on the slide, the wax supporting the tissue can be removed and the tissue can be stained using the dyes so that we can visualize the tissue structure under the light microscope. The solution that was used for this HNA staining are xylene, alcohol, and hematoxylin, tap water, acid alcohol, sodium acetate, and eosine. Then a drop of DPX mountain was placed on the slide and covered with a cover slip and dried overnight on the hood. So the next histopathology workflow will be microscope slide viewing. The instrument that we used for this procedure will be light compound microscope. After the slide has been mounted, it was observed under a light compound microscope that allows us to view the morphological structure of the liver tissue. The reason we choose this type of microscope is to observe the tissue cells under a higher magnification. Hematoxylin has a deep blue purple color and stains nucleic acids, while eosin is pink and stains proteins in a non-specific manner. Liver specimens stained with H&E will therefore typically display blue nuclei with the cytoplasm and the extracellular matrix in different shades of pink. So this will be the last histopathology workflow, which will be microscope, slide recording, and labeling. So for this, the instrument that we used is specimen logging form. Following that, the structure of the liver tissue was then labeled and recorded. Basically, these samples will be labeled on the frosted end of the glass slide with the patient's surname, phoneme, and the date of birth. So I'm going to summarize. Histopathology workflow of the liver tissue has a significant role in modern hepatology, which consists of fixation, tissue processing, embedding, sectioning, staining, and microscope slide viewing. The formalin that was used for the histopathology workflow to preserve the liver specimen by forming crosslinks and preventing degradation. The liver tissue is made up of several primary histological components, including morphological structure of the liver tissue sample. The liver histopathology workflow shows that in modern histology, a combination of several stain procedures is applied to improve the staining process efficacy. Last but not least, several stains have been modified and combined with other stains to improve their effectiveness in modern histology as a technique of improving histological stains. So that's all from us. Thank you.